Hello everyone. Today we are going to be talking about viruses. We have a few primary goals today, the first of which is to know and be able to describe the anatomy or morphology of viruses, to know and understand lytic and lysogenic cycles of viruses, to be able to answer the question, are viruses living or non-living and why? And finally, how do viruses cause disease? Now, you may recall from grade 10 science that every living thing is made up of one or more cells. Additionally, all living things have certain characteristics that they all do. So, for example, all living things can reproduce. All living things can excrete waste. They will also grow and repair. They're going to obtain energy in some way and convert it into a way that they can use it. They're going to respond to stimuli. They're going to move. And they're going to die eventually. What you see here in front of you is a comparison between prokaryotes and eukaryotes, two different types of cells. And no matter which organism it is, they're going to do those characteristics of life: excrete waste, grow, repair, obtain energy. They do it in so many different ways, but they still do it. There is an age-old debate about viruses and whether they are living creatures or not. Now, when you think of viruses, I'm sure that you think of disease or illness, and you wouldn't be wrong because many viruses are harmful to humans. Viruses are small, infectious, and considered non-living structures. They are not cells. They have no cytoplasm, and they do not display the characteristics of living things. They cannot grow or reproduce on their own. The only time they can do that is with a host. So like an animal or a cell. They do not produce or use energy. There's no metabolism, and they do not create waste. They are simply packages of genetic instruction, whether it's DNA or RNA, that enter cells and control them in order to reproduce. So the only characteristic of living things they have is the ability to reproduce, and that's only when they find a host that they can control. The goal of viral reproduction is to make copies so that they can then populate other host cells. It's important to know that antibiotics do not work against viral infections, only antivirals or vaccines. So, a virus particle is known as a virion. And we need to know some basic structure. And there's basically two structures that all viruses have, and sometimes a third. So the first structure is genetic material. This genetic material can be RNA or DNA. It can be single-stranded or double-stranded. If you recall, RNA is ribonucleic acid, and DNA is deoxyribonucleic acid. The difference between the two is largely the presence of an oxygen. So deoxy means missing an oxygen. Okay, the other component of a virion is the capsid, or head. So the capsid is basically a protein coat that surrounds the genetic material and protects it, and it's made up of proteins. Now, some viruses will also have an envelope. This envelope comes from the host. So what happens is, when the virus leaves the cell of the host, it can take some of the host's cellular membrane and wrap it around itself, and so then it becomes the virus's envelope. Now, we don't exactly know where viruses came from. We don't know their origin, but we have a few ideas. One popular idea, known as the progressive hypothesis, is that basically genetic material, or nucleic acid, somehow got the ability to leave the genome, or genetic material, of one cell and enter another. And so that's the progressive hypothesis. We also have the regressive hypothesis, which basically describes that viruses were once something similar to bacteria that became so dependent on their parasitic behavior that over time they lost the necessary genes in order to live outside of the host. Now, we've been talking about classification of organisms, but viruses are not considered to be organisms because they're not alive, generally speaking. So they're not included in our Linnaeus's classification system. But interestingly enough, they do follow a classification system that is similar to Linnaeus's classification system. It has similar taxa such as order, family, genera, and species, but it's a separate classification system. Now just below here, I have included the link of a BBC article from February of 2020, just describing how the new coronavirus actually got its name, because viruses have some naming standards as well. When we go to classify viruses, we do it based on a few different things: size, shape, and type of genetic material. Now, it's important to know that viruses are really, really small, like super small. If you take a look at these images, so over here, we have an animal cell right here. That's what that is. And beside this huge animal cell is a bacteria, which is considerably smaller when you're comparing it to an animal cell. And finally, over here, we have a virus. It looks like a bacteriophage to me. And this virus is much smaller than the bacteria. If you take a look up here, we have a few things going on. So our first image right here, we have a red blood cell, an erythrocyte, and beside it, a bacteria. If we zoom in, here's our bacteria, and down here, we can see a few viruses. If we zoom in again, we have a number of different sizes of viruses. So we have our smallpox virus, which is much larger compared to our poliovirus or our bacteriophage. Here's another bacteriophage, but these are two different sizes. And we have our tobacco mosaic virus. Shown here, we have a few examples of viruses. And now recall that I mentioned that viruses can be classified based on their shape. We have three shapes that we're going to talk about. The first is helical. Looking at these images, can you find a helical virus? We also have something called polyhedral. How about a polyhedral virus? Can you see that? Just guessing based on the meaning of the name. And the last one we're going to look at is complex. Do you want to take a guess? Can you see that? Okay, let's move on. So, like I mentioned, virus shapes. The first is helical. A helical virus basically has our nucleic acid, whether it's RNA or DNA, surrounded by a hollow protein capsid that's helical in shape, so it goes around and around, so it's helical. An example of that is the tobacco mosaic virus. Our second virus shape is polyhedral. Sometimes you'll see it called icosahedral. Now, polyhedral basically just means it is a geometric 3D shape with flat surfaces. Now, icosahedral is a polyhedral that has 20 sides. Now, many or most polyhedral capsids have 20 sides, like a 20-sided dice. And once again, we have our genetic information surrounded by our capsid. And the last shape we're going to be talking about is complex. where we have a polyhedral capsid. Within it is our genetic material, whether it's RNA or DNA, and that capsid, polyhedral capsid, is attached to a helical tail. So we have our helical tail here. This helical tail is largely made up of protein, and it helps the virus attach to its host cells. This structure is what bacteriophages have as a structure. A bacteriophage is a virus that specifically attacks bacteria. As I mentioned, viruses can have RNA or DNA, and there are some differences. Viruses with RNA often have a higher mutation rate. The reason for this is basically because there's less or no proofreading happening when that RNA is copied. We also have viruses with DNA. In contrast, these are usually more stable and constant. So because they're more stable and constant, we are able to create vaccines to help prevent viral infections. Some viruses with DNA include chickenpox, cold sores, genital herpes, hepatitis, respiratory infections. It's important to note that these are the names of the diseases, not the names of the viruses. But because they are stable, we can create vaccines. Take a look at this list right here. Do you have a vaccine against any of these? Now, if we look at viruses with, viruses with RNA, there's a higher mutation rate, which means it's harder to make effective vaccines against these viruses. So some viruses that have RNA are HIV, influenza, which is your typical flu virus, rabies, measles, mumps, and SARS. Now, SARS is an example of a coronavirus. SARS stands for Severe Acute Respiratory Syndrome. Now, the coronavirus that is currently causing our pandemic is also an RNA virus, specifically a single-stranded RNA virus, so there's only one strand of that genetic material. Another interesting thing is this influenza virus. Now, every year we come up with a new flu vaccine, and that's because there's always these mutations happening in the flu virus, and so doctors have to make an educated guess about which virus is going to be most problematic, and so they create specific vaccinations for those viruses. But there's so many influenza viruses out there. Okay, so we talked about how viruses are not usually considered living organisms, except for when they're replicating. Viruses can have two cycles, the lytic and the lysogenic. We're going to talk about both of them in some detail. Now, when I think of the lytic cycle, I think of it as being the more violent cycle. The reason for this is that the virus attaches to the host and injects its genetic information. So here we have a bacteriophage and a bacteria, and in the second step we have our bacteriophage injecting its genetic material, that's the blue thing right there, into the bacteria. The bacteria then the bacteriophage then controls the bacteria and tells it to create more viruses. So all the components required for a virus, the bacteria will then go, yep, I'll do it, I'll make it for you. And these components are put together and we get whole new bacteria. Eventually, this will cause the cell to undergo lysis, which just means that the cell membrane bursts and all the viruses are released. This can be a pretty fast process depending on the virus. It only takes about 30 minutes to produce 20 to 500 new copies of the virus, each of these copies going on to infect other cells. So the lytic phase, that's going to happen every now and then. The lytic phase is the more violent phase because our bacteriophages burst violently out of the host bacteria cell. Here is the lytic cycle in a little bit more detail. Over here I've included the steps. So the first thing that happens is attachment. So we're going to get a virus that attaches to a host. We're then going to get injection or entry. So it depends what it is. If it's a bacteriophage, then you simply have genetic information being injected. But if some other viruses will actually enter the host cell, so it just depends. We then have replication, so that genetic material and any required proteins are going to be made by the host cell. We're going to get assembly, and the new viruses are going to be assembled. And finally, they're going to be released through a process called lysis. I've included a few videos. If I can remember, I'm going to put them in the description down below. Okay, so if the lytic cycle is violent, then the lysogenic is a little bit more gentle. It's a little bit more subtle. So what happens is when a virus's DNA enters the host cell, what's going to happen is that DNA or RNA is going to be incorporated into the host's genetic material. Then, every time that host reproduces, it's also going to be copying that viral genetic material. If we zoom in on that picture there, we can see both cycles. We have the lytic cycle over here with our bacteriophage violently releasing from the bacteria. And over here, we have our lysogenic cycle. And what's happening is here we have our viral DNA or RNA, genetic material, which is then incorporated into the chromosome of this bacteria. When this bacteria replicates or duplicates, we are going to get more and more cells that contain that genetic information of the virus. What will eventually happen is that this viral DNA will separate, and then we can enter the lytic cycle. Now, this lysogenic cycle really just happens with bacteriophages invading a bacterial host cell. There's a slightly different process with viruses that are invading animal cells or plant cells. So here's the lysogenic cycle in a little bit more detail. Once again, first we have attachment. So right here we've got our little bacteriophage attaching to our host bacterial cell. We then have injection or entry, and so that genetic material is being injected. We have integration, so our viral genetic information is being incorporated or integrated into the bacterial genetic information. And then a state of dormancy occurs where this cell just reproduces, it duplicates, and we're going to get multiple bacteria with that viral genetic information. Eventually, at one point, there's going to be a triggering of that viral DNA, and it will return to the lytic cycle. That's that more violent cycle. If you check out my lesson online, there are a few additional diagrams. They're all showing more or less the same thing. It just depends on what you are most able to understand. So here's just a bit of a summary between the lytic cycle and the lysogenic cycle. So the lytic cycle is that violent one, where the virus destroys the host cell in order to release the viruses. When the lytic cycle is occurring, there are symptoms of a viral infection. So the host is affected by what is occurring in their cells. That lysis is traumatic. The cells will die. Comparatively, we have the lysogenic cycle, where that DNA or RNA merges with the genetic material of the bacteria, and this does not destroy the cell. There's no progeny, there's no extra viruses being created. It's just sort of hanging out in secret, hiding. And as a result, there are no symptoms of viral infection. We don't fully know all of the reasons that a virus may move from the lysogenic cycle to the lytic cycle, but there are things like environment or stress that can cause that to happen. This all leads to viruses that cause diseases. Now, if you take a look down this column, you will see many diseases that you have heard of. Rabies, HIV and AIDS, measles, mumps, rubella, polio, cold sores, herpes, chickenpox, hepatitis B. I'm sure that you are all familiar with them. It's important to know that the disease has a different name than the virus. So for example, HIV has a retrovirus. And there are specific virus names that are separate from what the name of the disease is. Similarly, COVID-19 is the disease, whereas we call the virus SARS-CoV-2. That's the name of the virus. SARS-CoV-1 was the virus that caused SARS. Recall that that's the severe acute respiratory syndrome that was prevalent in around 2003. So these diseases are caused by the virus attacking cells of the host. When it attacks the cells, we will see symptoms, and the symptoms are tied to what cells are being attacked. So for example, if you have a virus attacking the cells of the immune system, then you will end up with a compromised immune system, like with the disease HIV. Now, the effect on the host is not always immediate, especially if you have a virus in the lysogenic cycle or what is called a latent virus. We'll talk about that more in a little bit. Now, there's a certain type of virus called retroviruses. Retroviruses are really interesting because the virus incorporates its genetic information into the host's genetic information. So this is just like the lysogenic cycle that we talked about, where that genetic information is incorporated into the host genetic information. And the host will continue to reproduce its cells, and what this results in is a virus that remains dormant. So it's a little bit different because we're not talking about bacteria, but the host remains dormant for many, many years before the host ever develops symptoms. And an example of a retrovirus that you may have heard of is HIV. Now, HIV is the human immunodeficiency virus that attacks the body's immune system. HIV can remain dormant, but if it's not treated, it can lead to AIDS, acquired immunodeficiency syndrome. There is no cure to HIV. However, we can potentially eradicate it. If you're interested, take a look at the links in the video for how close to eradicating HIV are we. So there are some slight differences between lysogenic cycles and latency of viruses. Another way we call latency is persistent viruses. They're kind of subcategories of one another. So in a lysogenic cycle, a virus will specifically incorporate its genetic material into the genetic material of the host. whereas latency is a little bit more broad. It's basically the virus is hiding in some way from the host. As I mentioned above, that can happen in the case of retroviruses, where it also incorporates its genetic material, but that's not necessarily the case, and there are a ton of viruses out there. So some examples of latency that you would have heard of is the herpes simplex virus, or cold sores. Does anyone have cold sores? Often in the winter. I know that I get cold sores. And so what's happening is when you have visible sores, a virus is coming out of its latency to cause symptoms. And because I have cold sores, when I don't have cold sores, that virus is sort of just hiding. I still have the virus. I can still transmit the virus, but I'm more likely to transmit it when I have cold sores. And so it's a persistent virus because the organism, or me, will always have that virus hiding somewhere in my body. Another example is the varicella virus, or chickenpox. If you had chickenpox as a kid, you had the varicella virus. Now, this varicella virus is persistent because it can go into hiding and reappear as the disease shingles later on in life. So we've been talking a lot about viruses and how they infect hosts. There's 3 viruses that you kind of need to know about. Bacteriophages, which infect bacteria, and they look like that. Remember, that's that complex shape. We also have plant viruses, which infect plants, which would be this one here. And finally, we have animal viruses, which infect animals. And those are probably the ones you're most familiar with because they cause diseases that you've heard of before, like HIV, rubella, whatever. Now, when talking about the lytic or the lysogenic cycle, we talked about how a virus will enter or inject its genetic information. So when we're talking about bacteriophages, they're going to punch a hole in the cell wall and inject its DNA. So we have a picture of that over here. But this changes a bit when we talk about plant or animal viruses. So for plants that also have a cell wall, though made up of different things, viruses enter through tiny rips that already exist in the cell wall. And when it's animals, what can happen is the virus can enter the cells through endocytosis, which is where the cell engulfs the virus. Viruses are host specific, which means you will get one virus per a given host. And usually within that host, it's also a tissue type or a cell type. So for example, HIV attacks specific cells within the immune system. The reason being is that each virus has a capsid that is unique to their purpose. So on that capsid, they're going to have proteins that help it stick to its host. It's kind of like allowing, like a lock and key. So in order for a virus to stick to its host, it needs a specific key to fit into the host's lock. Now, I know that I just said that viruses are host specific. However, some species have similarities between them, which means that some viruses can infect several species. One example of that is the rhabdovirus that causes rabies. You can have birds, dogs, humans, lots of species can get rabies. Similarly, the swine flu can infect pigs or humans. Now, a topic of discussion has been, how do viruses spread? So this year, everyone has been washing their hands, wearing a mask, And the reason for that is because viruses can spread in air and water. So while we speak, or if we sneeze, then we're really seeing particulate droplets, which can contain viral virions, which can then be passed to those around us. There are some other things that can spread viruses, including insects, like mosquitoes, animals when they bite or scratch each other with rabies, and of course humans. If we exchange fluids, then we can pass on viruses. Vaccines are one way that we can prevent viral infections. These vaccines contain viral components or inactivated viruses that help our body to recognize and create antibodies that will then fight off the virus. It helps our body to recognize faster that there's a foreign thing in our body and to protect ourselves against it. These antibodies often stay with the person, allowing that person to remain immune to that virus, to that disease. There are some viruses that antibodies do not seem to be, the antibodies can be cleared from the body. So for example, our current COVID-19, these antibodies, there's still ongoing research, but these antibodies aren't forever, or that's what the research seems to show at this moment. So there are some viruses that you need booster shots for. So you get a booster vaccination to help recreate some of those antibodies in your body. Now, in the year 2020, people are actively trying to create a vaccine for the COVID-19 virus. There are a number of different types of vaccines. There's something called a live vaccine, where a very small part of the live form of the virus is injected into the person, though some of the harmful components are removed so that it's not dangerous. There's also something called the inactivated vaccines, where the vaccine is killed as much as you can kill a non-living creature, and so the virus can't replicate. They're often considered to be weaker, and so those booster shots are sometimes required with inactivated viruses. And there's one last vaccine type that I'm going to talk about, which is the mRNA vaccine, which is basically where mRNA is, or messenger RNA, is injected into the body, and the cells will use this mRNA to create proteins. These proteins can be sort of viewed as foreign objects, and antibodies will be created to recognize the proteins and can then attack it in the future. So this is one method that's being used to create a SARS-CoV-2 vaccine, because if you recall from the news, spike proteins are a distinguishing feature on the capsid of the SARS-CoV-2 virus. And so we're using mRNA to recreate these spike proteins that the body can then recognize and attack. Now, it is important to note that when this research was done, it was May 29th, 2020, so at this point it might be out of date. Just keep that in mind. Now, viruses aren't all bad. For one thing, viruses are important in killing harmful bacteria. So in that way, viruses can actually protect humans. Humans have also been making use of them. We can use, or we've been testing viruses to deliver a drug. This would be beneficial if we want to try and target specific cells in the body, because remember, those proteins will be targeting specific cells. We can also use viruses to genetically modify organisms, where we take a gene from one species and insert it into another species. Or we could use viruses for gene therapy, where we are using a virus to insert a new copy of a gene in order to help someone who may suffer from a genetic disorder. So that's our lesson on viruses. We talked about quite a few different things, including the anatomy of a virus, whether or not they're living or non-living, the shapes, the types of viruses, the life cycles, and diseases and vaccinations. As always, if you have any questions, let me know, and good luck.